this. Okay, yeah, and I was also involved in this NCI UMD partnership. And NCI is the National Cancer Institute, and it's a part of National Health Institute in the United States. And Jay was one of the group leader there. So they did most. He was also kind of like my advisor at that time, and there he worked. They work on the experimental part of the study, and we work on the simulation part of it. Of course, we did the computational. We have to the computational resources and the software that allowed us to perform this study. And so by the way, feel free to interrupt me if you have any questions. I try to give some background introduction, but if something's like not very clear, just let me know. I'm very happy to like provide more details. So if you are familiar with the central dogma of molecular biology, which tells you that all the information of a living system is encoded in the DNA, and during the transcription process, the RNA polymerase helps the information from DNA to be transcribed into RNA, particularly like the messenger RNA, and then the RNA interact. Sorry, any question?
No.
Okay.
I think you can continue.
So the RNA can then interact with the ribosome, and the information was then translated into proteins. And the protein can, in our living systems, like proteins are interacting with different molecules and interacting with each other to perform the downstream task. So this is like one direction process, and that tells you how the information from DNA flows into proteins. But however, this recent studies show that actually the real picture is way more complicated. So study shows that those small molecules can also directly interact with this nucleic acids. So there's a direct feed. So previously, when you think about biosignaling network, you always think about proteins interacting with each other, or protein interact with different small molecules. But recent studies show that actually this molecule directly interacting with the nucleic acids like the RNA to regulate the functionality of living system. And the system that we are particularly interested in is called the riboswitch. It's just a small segment of the messenger RNA. And by interacting with small molecules, that it can regulate the gene expressions, so control the expression of different proteins that control the functionality of living system. So here, I think there's a very nice review, talk about how this happens. So during the transcription process, the metabolites can bind to the riboswitch, and that will induce the conformational change of the system. So that will induce the forming of this structure called a terminator, will prevent the RNA polymerase from binding to this RNA molecule, therefore prevent the transcription process. During the translation process, without usually they will have the ribosome binding site RBS is exposed, so the messenger RNA can interact with ribosome to start the translation process. However, when the metabolite binds to the messenger RNA, it can also induce a conformational change. In this case, the ribosome binding site become buried. Therefore, the translational process will not happen. So this is a very like rough pictures of what's happening, and all this binding process happens at the abdomen domain of the messenger RNA, and that's the part that we want to study. Basically, we want to understand, like, upon the binding of a small molecule to the abdomen domain of the messenger RNA, here is the riboswitch. We want to see what's the conformational change what was the change that happens to the system, and want to understand what's the mechanism of this change. So in Jay's lab, they found that it's already well known that the cognate ligand PQ1 can bind to this riboswitch called the PQ1 riboswitch. And in Jay's lab, they found that actually they can come up with a synthetic ligand, which can also bind to a similar position, but with different, but that will induce different conformational change. So this becomes a very interesting system for us to study how small molecule interact with this this RNA system, especially like structural RNA. And that brings to the outline of my talk today. So I'm a computational chemist, so I work mostly on the simulation method. So I learned that the audience today are coming from different backgrounds. So I think it would be good for me to give some introduction about the methods that we are using. So the organization of talk, that my plan for this discussion today is like, I will first start with some introductions about the methods that I'm using and what is the challenge in there, and then the methods that we develop try to address these problems. And then I will apply them to this particular system, the riboswitch, and see how, and use that as an example to show how these methods can be used to address very challenging problems. And if time permits, I will talk a little bit about how we can use other AI methods called generative AI to start to generate molecular structures. But let's see if we have time for that. And so first, one thing that I would like to highlight before I start my talk, like for the first part, when I talk about the background, and also a part of the methods, these methods are not developed particularly for this system like ligand riboswitch interactions. It should be generally applicable to all the biomolecular systems that you want to study. So the tool we use is called, the computational tool we use is called the molecular dynamic simulation. The idea behind MD simulation is very straightforward. So you just need to know the potential of a system, which is determined by the coordinate of the atoms in the system. Then you can solve the Newton's law of motion. And since we are really talking about very, very large system, because for a typical biomolecular system, we are looking at thousands or even millions of atoms. So in order to solve these equations, we really have to use high performance computer resources. With that, we will obtain how the system's coordinate will change as a function of time. That's why you can see when you run the simulation, you will see a biomolecule. It's not just a static structure, it's something really that's moving around. So usually people call this computational microscope because it really allowed us to zoom in and see what happened for the molecular system at very high resolution. So for example, here we are looking at a protein that controls a type of protein called actin, and they are forming actin filament, which will control the movement and division of the cell. And we can, with this computational tool, we can really zoom in and see what happens at very detailed. And this understanding also allowed us to engineer the biomolecular system to develop new tools or materials that will bring real, like social, that will real bring like real social benefits. And other things I like to highlight is about the application of using these methods for drug discovery, because there's a law called the Ehrum's law, which tells you that it's become, so this the y-axis is how many money So with $1 million, how many drugs you can develop? You can see as time goes on, it becomes more and more expensive and also slower to come up with a new drug. Now the number is $1 billion and 10 years to come up with a new drug, but you should expect that now it's like even more and more expensive. At the same time, we know that there's the Moore's law, which tells you that the computational resource become cheaper and cheaper and more powerful. So the question we want to ask is that, can we use the rapid development of these computational resources to come up with, to bring us new insight about, to come up with new strategy for drug development, which can reduce the cost of this development process. I think many people argue that why we see such a slowdown of drug development. I think one reason is, from my opinion, is like actually we really lack enough understanding of how these small molecules interact with the biomolecule, interact with biomolecular systems. And with the help of computational methods, I believe we can use this as a new tool to bring us new insight, and therefore completely change the way how we develop drugs and come up with and use this as a pathway to what we call the computational drug discovery, instead of traditionally the experimental drug discovery process. So everything looks nice about MD simulation. It seems that it's very powerful. It can solve many problems, but one thing we should realize that this is not a perfect tool, and there's a big challenge that we are facing when we run molecular dynamic simulations, and we usually call this the real event problem, or how to sample the real event. So what do I mean by the sampling real event problem? So when we run the molecular dynamic simulations, we have to evolve the system at femtosecond time scale, so which is 10 to the minus 15 seconds. So every step we can only the system forward in time by this amount. And with very powerful computational resources like high performance computer clusters, or people also develop like chips that can only be used to run MD simulations, we can at most reach around millisecond time scale per day. However, the process that we want to study, for example, the protein conformational change, so this is kind of like the protein folding, and also the protein ligand interactions, also the RNA ligand interaction process happens at the minute time scale. And there's some even slower process like nucleations can happen at several minutes, or even at a time scale of several minutes and even hours. So therefore, there's a huge gap between what we can do with today's most powerful computational resources and the process that we want to investigate. And in order to give you a sense of how big this gap is, let me give you a very concrete example. So this is a system. This is a system called actin filament. This is an actin molecule, and here we want to study how this red small molecule is a phosphate, how it leaves this binding pocket. So the time scale of this from the experimental measurement is around 1 minute. And now assuming we have 4 very powerful GPUs, we can only run 50 nanoseconds per day. With this, we can only evolve the system forward in time by 15 nanoseconds every day. And the simple calculation will show that in order to finish this calculation, it will take like 10 to the power of 9 days to finish the calculation. So this is not a back number, but just longer than human history, and no one has the patience to really run simulation for this long. And also I would like to highlight that actually there's a huge computational cost associated with this calculation, because if you are familiar with, if you heard about like ChatGPT, in order to train the models with the same amount of computational resources, it will take like 10 to the power of 4 days to train the model. So if you got the impression that training this large language model like GPTs are very, very expensive compared with molecular dynamics simulations, they are just nothing. So that's the questions that the whole community is trying to address. That is, with the given computational resources, how we can really accelerate the simulation to make sure that we can sample or study the process that we want to investigate. Hope that is clear. Just feel free to jump in if you have any questions. So basically, I would like to show a plot. I would like to show is like this. So the x-axis show the time scales, and the y-axis show the size scale. So with currently available resources, we can only simulate the system that lies below this curve. So we can only simulate system with very small size. We can only simulate system that simulate a process that happen at very short time scale. And however, the things that we are interested in are really lies above this curve. So we cannot just do like simple molecular dynamic simulation to investigate these systems. So the question I want to show is that with the help of machine learning, we can really accelerate the simulation to make sure that we can simulate the system with larger size. And we really want to push the boundary of the simulation we really want to push the boundary I showed before to make sure that we can simulate systems, simulate the process that happens at a longer time scales. So that's the problem that, as I mentioned, so in order to study any like process, including the ligand riboswitch interactions, we really need to have methods that help us to overcome the timescale limit. And from now, I'm going to talk about a particular method called RAVE that we developed try to address this problem. As of course, it's a AI-based method. And I would like to hope that I can give you some sense about like why AI has become particularly useful in this scenario. So the first questions we want to ask is like, why the simulations?
Dr. Wang.
Mm-hmm.
Yeah, I had a question about the previous slides. Is it true what the previous one?
Okay.
Yes, this one. The
Mm-hmm.
simulations, as the molecular size becomes bigger, simulations become hard, and it is easier for smaller molecules. Is that what this graph is portraying?
Yes, yes. I think you are correct. It's like if the system becomes smaller, it's easier for us to simulate because it takes less time for us to evolve system forward by one step. So because each time you need to really calculate the interactions between all atoms. So as the system becomes large, you have so many atoms, and you really have to calculate the interactions between all of them. That's why it becomes slow. That's why, as you say, there's a trend that as the system becomes bigger, the time scale you can reach becomes shorter and shorter. And that's why we really want to push the boundary and see if we can do better in terms of both run the simulation for a longer time and also investigate system with larger size.
Okay, thank you. Thank you for the
No
clarity.
problem. Mm-hmm. Yes, thanks for the questions. And yeah, and the question we want to understand is like why the simulations are so slow. So if you are from like computational chemistry, you might familiar with the concept of the reaction coordinate. So, but here I think I still like to give a brief like introduction about that. Basically, it's a variable that we are used to describe the reaction process. So the reaction process can be something like quite arbitrary, depends on what's the process that you are interested in. And for example, like if I'm interested in the protein folding problem, so the reaction coordinate may describe how a protein move from an unfold state to a folded structure. And usually, and the y-axis I'm showing is the free energy. So the system always like to stay at a low energy state, and it's moving around because there are thermal fluctuations. The reason why the simulations are so slow, because it's that it takes a very, very long time for the system to gather enough energy to cross this energy barrier to move to another state. So if you run a simulation, you will see that the system always stays at one structures and then just vibrating locally. And there's only a very, very rare chance that it gather enough energy to across this high energy barrier. And the chance of crossing this barrier is proportional to the exponential of this barrier height. So as the barrier height increase, the chance that the system across this barrier decrease exponentially. So that's why we have the real event problem. And the things we want in order to accelerate this reaction process, because that's the process that we want to investigate, we really have to help the system to cross this barrier and visit other states. And similarly for the riboswitch system I mentioned before, so you can think about one state is the ligand bound state, and the other state is the ligand unbound state. And similarly, in order to simulate this process, we really want to help the system to cross this energy barrier. And what can we do with that? And usually, traditionally, we have a group of methods called the enhanced sampling method, which can be used to address this problem. And here I'm going to give you two examples. But actually, there are many, many methods like this. But here I would like to just give you two examples to give you a sense like what's happening. So this is what happened in a normal molecular dynamics simulations. Without doing anything, you will see that the system just being trapped at one energy minimum. And to overcome this energy barrier, you can do something called the metadynamic. The idea is that during the simulations, you can keep adding some external potential during the simulation process. And it's like in these simulations, the external potential is added at a place that has been visited by the system. And as this external potential keep accumulating, the system will gather enough energy to cross this energy barrier and visit other new states. And that's the rough ideas of metadynamic.
Doctor, I'm wondering whether the crossing the energy barrier has anything to do with the computational power, or it's about since the bias can improve crossing it faster, but does it have anything to do with the computational power limitations?
So could you repeat your question? So I'm a bit confused about.
My question is about when you look at the two graphs here on the normal molecular dynamics, but then when you introduce metadynamics by adding a bias potential, we are seeing that the reaction quickly overcomes the reaction barrier. And I am asking, is crossing the energy barrier, does it have anything to do with increasing the computational power, or computational power doesn't have anything to do with it?
Yeah, the reason why I say this is like in order to that's the exactly problem that we want to address, because if we just run normal molecular dynamics simulations, we have to stay here forever, and that means we have to use a lot of computing resources to wait the system until it across this energy barrier, and that really takes a long time for this to happen. And here you can see that it crossed the barrier way more faster than the normal molecular dynamic simulation. Therefore, we can save a lot of computational resources, because as long as we see the system visit these two states, we can stop the simulation. The simulation is done. And usually the things happen on the right can finish in a few days, and on the left may take many, many years. So that's why, and of course, if you want to do this kind of like metadynamic simulation, you have to take extra amount of computational resources, because you are doing something else beyond just doing the normal molecular dynamic simulation. But the cost of this actual calculation is negligible. It's just, usually we don't care about that at all.
Wow. Thank you.
Mm-hmm. And analogies I like to make here is like, yeah, you might heard about a story about a smart donkey in order to try to escape from this well. It step on the sands that is thrown into the well, so that helps it to escape from this like energy minimum. So the idea is the same. It's just very straightforward and easy. And the other method is called weighted ensemble sampling. So here, we need to do is to divide the reaction process into different stages. And here we can see that we divide this process into 3 stages. They call it being 1, being 2, and being 3. And there they want to make sure that for each stage, there are enough simulations in order to, because you want to do like statistic of this simulation process. So you want to make sure that there's enough samples or data from each stage. And here, let's assume that I want 2 simulations for each stage. So what they can do is like at the very beginning, you have just 1 simulations. As I said, I want 2. So I can split or copy these simulations into 2 and just let the system evolve forward in time. And at some point, you will see that 1 simulation stay at the stage 1 or being 1, and the other simulation goes to being 2. And as I said, I want 2 simulations for each stage. So I can split each simulation separately. So I got 2 simulations for stage 1, and then 2 simulations for stage 2. As system move forward, there's a good chance that one of the simulations go back to the original stage. And there are too many of them. I can randomly choose one of them and then just terminate the simulation. Therefore, I maintain 2 simulations for being 1. And similarly for being 2, there are not enough simulations. I can again split that into 2. As the simulation keep moving on, you can keep doing these operations. You will end up have 2 simulations on each region. And therefore, it's kind of like you are trying to push the system forward. So every time the system makes some progress, you replicate the simulation to make sure that it provides enough, it's running enough simulations or computations on the region that we're interested in. And therefore, you can gradually populate the whole energy landscape to gather information about this system. So my understanding, my way of thinking about this is like a few blind people, in order to get the understanding of the whole elephant, each of them can touch or measure the different part of this big elephant. And by exchanging information between them, they can get a sense like how the whole elephants look like. There's the methods called weighted ensemble sampling. And all these methods looks nice and has been used to solve many challenging computational biomolecular problems. However, there's one limitation that I would like to point out, is that all these methods relies on the situations that we know what is the reaction coordinate. So we have to have some low-dimensional discrete way to describe how the reaction happens. And this is actually not a very simple task, because for biomolecular systems, the energy landscape is usually, you are talking about a system with thousands or millions of degrees of freedom. And the energy landscape of these systems can be very rugged and very complicated. And what you want is to find meaningful representations that when you project the system from the original coordinate, to R to the some simplified representation S, you will get a smooth but also meaningful energy and surface that can nicely capture the reaction process. So let me give you a very simple example. So this is a 2D system that I want to investigate. And what I'm plotting here, that the contour I'm showing here is is energy profile. So for the blue state, for the blue region is the low energy region where the system want to state. And the yellow region are the region with high energy where the system don't want to visit. But in order to go from one blue region to the other blue region, you really need to cross the high energy barrier lies between them, because that is indicated by the yellow lines here. And now the text is to how we find a one-dimensional reaction coordinate for this process. First, you can choose the one as I show here, shown by this blue line. This is not a good choice, because if you project the system along this blue degree of freedom, this is what the energy landscape that you will see. It's bad in the sense that it doesn't tell you anything about this original system, because you can see here there are just one basins. But in the original system, we know that there are two energy minimum. So in contrast, this the one I show on the right is a better one, in the sense that if you project the system onto these directions, you will get a more meaningful energy landscape. This really tells you that the system has two states, and in order to move from one state to the other, there's an energy barrier that you need to cross. And the success of the previous enhanced sampling methods in order to successfully apply the enhanced sampling methods that I mentioned, you really need to know this right projection of the system. You need to choose this good reaction coordinate. And that's a problem that that's a problem that without the AI methods is very hard to address. So the reason why we think AI is very powerful is by realizing that AI is very, very good at learning from high-dimensional systems and figure out what's the most important features. For example, like if you this is not a very this is old news, but what I want to show here is that AI Go is a very complicated game. It's also a very high-dimensional thing, and AI can really learn from these high-dimensional structures and identify which state is better than the other one. So similar to the case I mentioned before, we want to find a reaction coordinate that's better than the others and use that to perform enhanced sampling. So that comes to the AI methods that I would like to discuss. It's called the RAVE, and let me again mention the task what I want to do. So looking at a very complicated biomolecular system, I want to come up with simplified pictures of of this process that can be used to describe the reaction. So that's what we call the reaction coordinate. And analogies, here we are defining the reaction coordinate as the most informative degree of freedom. And I will discuss in more detail about what I mean by most important degree of freedom. But let me look at the case I'm showing here. This frog is trying to catch this flight. So a flight itself is a very high dimensional. It has its own color. It's making some noise, and it's made up of many atoms, molecules. But somehow the brain of this frog is very efficient. It realized that in order to predict the position of this flight, in order to catch it, you need to focus on its velocity and position. That's the two information that you need to know. So therefore, this gives us the inspiration that the biological neural network are very good at identifying important features from your signal. So imagine that you are getting a very high-dimensional signal, which contains some useful features. And also there are some lots of noise. And the human brain or the frog's brain or the animal brain are very good at extracting what's the most important features. So, but this is done by a natural, a real brain or natural neural network. How can we do this for an artificial neural network or AI system? Based on this idea, we developed a method called reweighted auto-variational Bayes for enhanced sampling, or in short, you can just call it rave. And rave in English just means like a dancing party. And so here you can think about, we can describe this as use math and physics. So think about a case, we have a very, very high dimensional signal X. So that's what we observe. So in the case of the frog trying to catch the fly, it's all the information about the fly. And in the case of like molecular simulations, this can be the coordinate of all atoms, how the system look like at a particular time step. And we want to find an encoder, which encodes the system into some low dimensional representation S. And this is the information that we want to learn. And in this case, there's always like different choice. You can encode the system into something very complicated. You can also encode the system to something very boring. So there are many, many choices. However, what I want is that given these low dimensional representations, I want to decode the information back to some high dimensional information. So, and what I want is that the high dimensional system should capture useful information. For example, in this case, I asked the system to predict what is its next state given this low dimensional representation. So this can be phrased in physics, use the information theory, which can trace back to like Shannon. So basically what we want, first, as I mentioned, I want the system to encode the information from this very complicated system. I want it to forget information that is not very useful. Therefore, I want to measure the I, which is the mutual information between these two systems. If the mutual information between these systems these two representations are very low, that means there's very little information that shares between them. At the same time, I want this to make as I mentioned, I want to use this information to make a prediction. Therefore, I want to maximize the mutual information between these two. So you can think about there's a trade-off here, because if you forget everything about this current state of the system, there's no way you can make any good prediction. In order to make a good predictions, so you really need to measure, or you really need to capture or identify the most useful information that can be used to make this a prediction. So you can write it down as a trade-off here. You want to minimize the first terms, as I say, you want to compress the information, but at the same time, you want to minimize you want to compress information, but also you want to make good predictions. So there's a natural trade-off here that can be done by maximizing this objective function. So you want to maximize the first term and minimize the second term. And this can be done if you do some tricks. If you look at this deep variational information bottleneck paper, you will find that you can actually rephrase this as a problem of a neural network, which has an encoder and a decoder. And by training a neural network like this, so you take the current state of the system, you first encode that into some low dimensional representations, and then you use that decode that into predict the future state of a system. And by training this neural network, you will learn some meaningful representation of the original system as here, and we can use this as the reaction coordinate. To give you a more concrete example, so I have the current state of the system at time t. I want to encode that into some low dimensional representation, which characterize the many states of the system. And then I want to decode that back into predict the future state of the system. And this latent representation is learned automatically by the AI algorithm. That's where we take the power of AI, because it can really look at this very high dimensional object and identify what is the most important low dimensional representation of the system. And if you are familiar with machine learning methods like VAE, rational autoencoder, the idea is kind of very similar. So it seems that, okay, now we have very, very good tool to learn or identify what is the reaction coordinate. The problem has been solved. However, unfortunately, this is actually a chickens versus egg problem, because, as I say, if you know, I would like to think about like this kind of sampling or simulation problem is kind of like you want to work out of this maze. And those enhanced sampling methods is kind of like the tricks that allows you to directly across the wall. So that will help you to escape or work out of this maze very quickly. But that only happens when you know what's this direction. That's why we want to learn the reaction coordinate. However, if you, at the very beginning of the simulation, you just sample some very local information, and this information is very limited. Given those very limited information, it's very hard for you to understand the whole energy landscape of the system. So therefore, it's very, very hard to make good decisions about what's the directions to move. And and here that's why we call this chickens versus egg problem. So as I mentioned before, in order to perform enhanced sampling, in order to save computational resources, we really need to know what is the reaction coordinate. However, in order to know the reaction coordinate, we need to sample the system very well. So it's like we want the chickens and eggs, but we have none of them. So that's the problem. To address this problem, that's the whole training framework that we have. So first, we start with a simulation. It can be just like the unbiased, very limited simulations I mentioned before. And by running these simulations, you just obtain some very limiting data, but you can still apply the machine learning methods to learn what's the meaningful information from this very limiting data. And then you can use that as a reaction coordinate to sample the system. And by running the enhanced sampling methods I mentioned before, you can collect a little bit more data that allow your machine learning methods to learn again to collect more information. And based upon of that, you will learn a better reaction coordinate, and you can perform better enhanced sampling, and you collect better data, and you keep learning. So this is like an iteration process. The AI methods trying to keep generating new data, learn from it, and use the new information to generate even better data. So as this process keeps going on, you will finally learn a good reaction coordinate that capture the reaction process. And at the same time, you also already generate a lot of data from this system that you want to investigate. Okay, so I have applied this to some like different systems. So this is kind of like a time series of the simulation process. At the very beginning, you can see the value doesn't change a lot. They just stay at one position. But as you keep iterating, you are learning better reaction coordinate. You can see there now you can see transitions between different states that really allow you to sample this energy landscape way more better than just unbiased molecular dynamic simulations. And actually, if you want, there are some methods that allow you to not only obtain the free energy difference of this system, you can also obtain the timescale behavior, the kinetic of this system. And at the very beginning, we apply this method to a relatively simple system called RNA dipeptide. So this is kind of like the most simple system that we usually test as a benchmark, and also this T4 lysozyme, because this system has been well studied, so we use this to benchmark our methods, and it worked very well. And with these methods, we can largely reduce the computational cost associated that we need, because usually so even for a system like this, it's very hard to obtain anything useful with just simple traditional molecular dynamics simulation. So okay, now we can go back to the riboswitch case that I talked about. So it's a system it's very hard to simulate. So here I run a 2 microsecond unbiased, just basically you can think of this as naive molecular dynamics simulation. You can see the both ligand stayed at a binding pocket and nothing really happened. But 2 microsecond unbiased simulation is short in terms of like the time is like just 2 microseconds. But however, to run this simulation, I really want the simulations on high performance computers for many weeks. From there I already collect some useful information, especially regarding how the system fluctuates. And this can be, so here what I showed is that I'm comparing my simulation result with the experimental measure. There are ways to measure this. This can measure the fluctuation of the system at the residue level. So basically, our experimental collaborators use these sharp methods to measure how each residue fluctuates. You can see here, for example, like this part really fluctuates a lot, and there are some part which is more rigid. Basically, this tells you some like property of this system. You can see that there's a good agreement between our simulation methods and experimental measurement. And because the reason why we care about this is like actually the simulation of RNA system is way more challenging compared with simulation of protein system, because protein is something, the force field for protein is very well developed. However, for system like amino acids, the force field is really not that good. But here we are very happy to see that there's a good agreement between the two, suggesting that the force field is okay, and we can use that to investigate this particular system. And as I say, if you just run unbiased simulations, you will see nothing. It's very hard for you to see this unbinding event to happen. However, with the method wave I just discussed, we can really use that to accelerate the simulations. And in order to run this, if you just use unbiased simulations, it will take more than 2 years. And now with the AI methods, we can finish this in just a few hours. And one thing we found to be very interesting is like, actually we found that the unbinding happens through 2 different pathways for these ligands. And we can look at this at a more quantity level. So here I'm plotting the free energy of these 2 systems. So the x-axis I'm showing the coordination number. So the coordination number describes the level of contact between the ligand and the RNA system. So when it's bind at the binding pocket, there are lots of contacts. So the coordination number should be very high. And when it really dissociates, there will be the contact will there will be less contact, so the coordination number should decrease. So you can think about the dissociation happens by moving to the left of this panel. So here are the 2 bound states, and when it really unbound, you need to cross this energy barrier. And what's nice is that we can measure the energy difference between these 2 systems. And from experiment, we know it's very hard to measure this, but our experimental collaborators have been come up with very precise measurement of this free energy difference, which is around 3 kcal per mole, and which shows that the cognate ligand is way more stable than the synthetic ligand. And from our simulations, we also reveal the same finding. So this is a way to highlight that this highlight that with simulation methods, especially with the help of machine learning and enhanced sampling methods, you can obtain a lot of useful information from the system that you want to investigate, and that really align well with the experimental measurement. So that's this just one step. So can we do better? Because now we run the simulations and we believe that we obtain many important insights of these systems. So what can we do with this insight? So we have simulations. Can we come up with a way to help us to understand what is the important interactions that happens during this process? And this knowledge of this, can we use this knowledge of interactions to come out with some new experiments? Basically, so we want to use these methods as hypothesis generators, and then where we can use experiment methods to confirm this hypothesis, and therefore this like collective behavior will help us to obtain new understanding of this system. So what we can do is this. So here I would like to give you an example. So here I'm looking at, as the coordinate number change, how the system fluctuates. And here I'm looking at a system at the residual level, because that's really the advantage of simulation. It really allowed us to look at the systems with very, very detailed information. So for example, here you can see that for this residue 19, nothing really changed. However, for some other residue, like residue 1 here, you can see there's a big change for both systems. And more importantly, the change is very different across these two systems. So with the cognate ligand, it's actually way more flexible during this process. And for synthetic ligands, it doesn't change a lot. From this, we can hypothesize that we can suggest that probably this is a more important residue that's associated with this ligand unbinding process. Actually, we do very thorough investigations. So let me move on. So from there, we suggest 2 residues, and we think these residues play a very important role for the unbinding process. So which one is the residue U22 and the other is A33. And because other observation is that for the cognate ligand, the unbinding really happens through this top pathway, which is closer to this U22. So we suggest that if we mutate this residue, we should have more significant impact on the binding behavior of the cognate ligand. However, for the synthetic ligand, the unbinding happen more frequently through this bottom pathway. So we suggest that if you mutate this residue at the bottom, there's a good chance that you have a higher impact on the binding behavior of the synthetic ligand. And this is what we suggest, and our experimental collaborators really do the experiments, and their results perfectly suggest they use some methods called micro-scale thermophories, which is something which I really don't know what's happening there, but the conclusion is that their measures agree perfectly with our simulation suggestions. So so basically what we want to show here is that the simulation tools can not only provide us some new insights about a system. It can also it can really go beyond just like validating experimental measures. Instead it can really give us new insight, which can help us to inform what's the next new experiments that we should do to better understand the system that we want to investigate. And I would like to, so one thing that ends my introduction about how we apply this AI methods to study like very, very challenging systems, especially as the case of riboswitch I just mentioned. We can use that to study the interaction mechanism between ligand and nucleic acid. And based on this, actually, I have applied this RAFE methods not only to the ligand RNA system I just mentioned, has been applied this to many, many very, very challenging systems, especially like this one. So without any AI methods, you need to more than 10 to the power of 9 states to finish the simulations. But now with AI, with the help of AI and enhanced sampling methods, we can finish the simulation of this within just a few days and collect lots of informations. And if you are interested in playing around with these methods, and of course, and also I want to say that this method has really been updated. We have many new versions of this method to try to improve its performance. And you can visit this GitHub page where we have all the code being open source, so you can feel free to use that. And if you have any questions, definitely feel free to reach out to us. So with that, I'd like to summarize what I just said before. So first, AI is a very powerful tool for us to address challenging problems for biomolecular system. By combining these methods with ideas in thermodynamics, we can really, and statistical mechanics, we can really use that as a tool for us to investigate the property of challenging biomolecular systems. And in particular here, today I show a method called RAVE, which by combining this with enhanced sampling methods, we can really we can address the timescale issues that we are facing with the molecular dynamic simulations. We can move from the very limiting microsecond timescale to much longer timescale, like minutes or even hours. And in terms of applications, here today I discuss how we can use these methods to study a very interesting system called riboswitch, and how it interacts with small molecules. And that is really not the limitation of this method. If you want, you can really apply this method to study different type of systems and use that to understand how biomolecules interact with small molecules. Yeah, so how I'm doing at time. So how many, okay, I'm already used the 1 hour, that's good. So any question?
Thank you, Dr. Wang, for this wonderful and technical work. Yeah, I think for me, the only question I have is about the generative AI component. Yes, yeah, we see that Rev is using at the core variational autoencoders, as you would say. And you did this work, I think, like 2 years back.
Yeah, exactly.
Before the explosion of diffusion models.
Right.
But if someone is to think about it in form of diffusion models, what sort of approach would you take? Or would you take the same approach? Or would even diffusion models be really an application here?
Yeah, I also published paper using like diffusion model to apply the sampling probe. Actually, I have a slide for that. So yeah, I think now there's a trend of using diffusion model to address this problem. So there we are more thinking about, let me see, we can use diffusion model to, let me see, sorry, let me move on. Yeah, we can, similarly, this is also, actually I should go, yeah, this is also RNA system. And you can use diffusion model to learn from the simulation data and use that to generate like new structures. So because that's what diffusion model can be used to do. And this is also, I think our group might be the first group that use diffusion model to generate like molecular structure. And this field has really like emerged a lot. People has used that to study many very challenging systems. Even now, I'm also working on something like we can use this to, because one thing interesting about diffusion model is that it can generate samples that follow some constraint. So here we are showing that given some constraint like cosquare model, we can use that to backmap to all atom structures. So here we are trying to do something even crazy, like we try to use given some like cost representation of HIV capsid. So now I'm not talking about just one protein. I'm talking about over 1,000 proteins. We can use that to backmap all atom details of the whole HIV capsid. So there's many opportunities in terms of using like diffusion model. But one thing I would like to point out that it's different from diffusion model and the approach I mentioned is that diffusion model can, at this time, more or less, mainly being used to study the distribution of structures, but that doesn't contain any information about the kinetic or the dynamic of the system. And the methods I mentioned, the wave, can be used to study like how fast system move between different states. That's an advantage that I would like to say about the using some methods like wave and combine them with more traditional enhanced sampling methods, because there's a better chance that we can like obtain other useful information about the system. Yeah.
Wow. Thank you. That's an interesting take to look at this work. I think for me that's all I had. And when you come to think about variational autoencoders, I had not really imagined them in this work. And actually, your work is I've not seen much work about variational autoencoders and binding sort of prediction or binding affinity. Yes, so it is really an interesting look, and really I want to thank you very much about the presentation.
Yeah, we're happy to come and discuss this.
Yeah, so from my group, I don't know if anyone has a question, and if nobody has a question, I really want to release Dr. Wang. Yes, thank you very much. Looks like nobody has